Next on our program is a keynote talk from Talia Karasov. And let me give you a little bit of background. So Talia is one of our newest faculty. She and Michael both just arrived, I don't know, last week, I think. Talia was born and raised in Madison, Wisconsin. She was an undergraduate at Stanford University, and she worked in the lab of Dmitry Petrov, where she studied evolution of insect resistance in Drosophila, and then looked to see how this resistance spread through populations. This is kind of a theme in her work, looking at how looking at resistance and how things move. She then went to the University of Chicago. She joined Joy Bergelson's lab group, where she started working on plant pathogen interactions. And what she did is she used Arabidopsis and looked at coevolution between Arabidopsis and a major pathogen. She then went to do a postdoc with Detlef Weigel at the Max Planck. She continued working on plant microbe interactions, but in a much more, in a very natural context, and trying to understand how plants and their pathogens co-evolve. And then also understand how both plant genetics and the plant microbiome influence the emergence of pathogens. Now, many of you will remember that she came and gave a talk in 2018. She gave a talk for the LARC, she gave the LARC lecture at the Science Retreat. That's where many of us absolutely fell in love with her research, and we're so happy to have been able to recruit her here. In a moment, I will turn this over to Talia. And during Talia's talk, just as in the others, when you think of a question you'd like to ask, please enter it in the chat. And after her talk, we will go through at least some of those questions. And let's see, Talia, are you here?
I am here, and I will share my screen.
Thank you. We're so happy to have you, and I will turn it over to you.
Okay, how does that look? Do you see a functional screen now? Good.
Looks great to me.
Okay. Hi, everyone. It is really great to be here today. Before I start, I wanted to thank the organizers. This has gone without a hitch, and it's actually been pretty fun. I was smiling for a lot of that trivia, and the talks have been really good. So it's a pleasure to be here. So I am going to be talking now about the work I've done over the past few years in my postdoc, and then with just a little taste of what I foresee in my future, continuing the similar ideas to what I'll talk about today. And this work is focused, as Leslie said, on host pathogen interactions, primarily plant pathogen interactions, and the evolution of these interactions. Studies of pathogen in agriculture have revealed a common pattern. This pattern is one of clonal expansions, in which a single genotype of pathogen emerges within a host population and then spreads widely within that population and then to other surrounding populations. To give you a sense of this phenomenon, I'm showing you one representative example in which the authors studied the spread of the bacterial pathogen Pseudomonas in global agricultural tomato populations over a 50-year period. And what the authors found was that at any point in time, tomato, even on different continents, were colonized by basically the same genotype of pathogen, with every few decades a new lineage of pathogen emerging and spreading globally. Now, what is it about perhaps agriculture or these plant populations that enables a single genotype of pathogen to spread so effectively and widely? Now, of course, the answer is going to be complex, and it's not just going to be one silver bullet, but we can posit a few of the possible contributors to this. So one is that of low intra-species diversity. So modern agriculture, frequently we plant large swaths of land with only a single genotype of the crop, so single genotype of tomato, for example. So if a pathogen is able to overcome the defenses of that specific genotype of tomato, it can colonize all the other tomatoes in that field. In the same vein is low interspecies diversity. That tomato is not surrounded by other species, but is surrounded by a number of other tomatoes, such that transmission between plants is very rapid, and that the microbe can colonize all of the hosts in a single field or across a region. And thirdly is low environmental heterogeneity. And when I say low environmental heterogeneity, I say it ambiguously to encompass both biotic and abiotic environmental heterogeneity. So we know that much of the treatments that we do to soil can homogenize both the microbes that are there, so pathogen suppressing microbes, but also the nutrients are more constant than would be normal in wild populations across many agricultural plantings. So what is happening in a paradigm in which we have greater diversity at probably all of these levels? What is happening in wild plant populations in which we know that there's extensive genetic diversity within plant species? Actually, a fair amount is known about immune diversity even within species, and that there is extensive immune diversity. There's more than one plant species in a population. And then we also know that there are micro niches for environments that are more diverse in these wild plant populations. So the projects I'm going to tell you about today are centered on this larger question of, when we look to these more diverse environments, how are pathogens evolving and spreading in these wild populations? And the first project I'm going to talk to you about is asking this question on a very basic level and just trying to get a snapshot for one model system to see how the pathogens are evolving and spreading. And then the second part, it's the initiation of our efforts to try to find the relative contributions of host and microbial genetics and the environment in dictating the spread of pathogen. So to begin to address this question in a tractable manner, we decided to focus on a host pathogen interaction in which we could know something about the ecology of the host and the microbe and about the genetics. So we utilized the resources available for what is for now the model plant species Arabidopsis thaliana, or at least the model molecular biology plant species for Arabidopsis thaliana, for which we know about its global distribution. It is found in many locations in many types of environments. And there's also extensive genetic resources for Arabidopsis thaliana. And then for the microbe, we decided to focus on the bacterial genus Pseudomonas. So why Pseudomonas? So Pseudomonas has hundreds of genotypes or strains that are known to be host specialized on different crop species, inducing large economic losses on different agricultural crops. But also we know that Pseudomonas is a natural colonizer of Arabidopsis thaliana. So here I'm showing you the metagenomes of the leaves of Arabidopsis collected in Southwestern Germany, which is where I did my postdoc. Each column is a single plant, and this is just a snapshot of the composition of microbes that colonize the leaves of Arabidopsis. And what I want to draw your attention to here is this Pseudomonas. So Pseudomonas is among the most abundant genera in these Arabidopsis that we collected in Southwestern Germany. And from other phenotypic testing, we know several of these Pseudomonas strains that you can collect from Arabidopsis have a pathogenic phenotype on Arabidopsis. So what our aim was to take this interaction between Arabidopsis and Pseudomonas and go to local populations in the area. This is where I did my postdoc in Tübingen, Germany. Go to local populations and take snapshots of the microbes that were colonizing Arabidopsis in these local populations. We got, in collaboration with a researcher at the University of Tübingen, we together collected information from these populations, from 6 populations over 3 years and 2 collections per year. So we had spatial and temporal information about the spread of pathogen. And from these populations, we took several pieces of information. So we went to the field, we collected plants, we focused specifically on microbes that colonized inside of the leaves, so foliar pathogens. We cultured widely. We cultured over 3,000 Pseudomonas from several hundred plants that we collected from these fields. And then we also took other pieces of information about the state of the plants. So to assess which microbes were present within these leaves, we did traditional microbiome analysis or amplicon sequencing of the 16S locus. So what this tells you is about the composition of species or subspecies of microbes that are present in your focal sample. So we classified these species, subspecies at the level of operational taxon unit, or OTU. I will use this term throughout the talk, so just remember OTU is like something like subspecies. To get a sense not just of which microbes were there, but how much of them were there, we actually used shotgun metagenomics, where we looked at the ratio of plant reads to microbial reads, and this gave us a rough idea of how much of the microbe were there, how successfully they colonized the plant. And then to understand how the microbes we collected related to one another, whether we were seeing these kind of clonal outbreaks, we took 1,500 of the isolates that we collected from the field. We developed a very cheap method of whole genome sequencing for these isolates. So we got 1,500 full genome sequences from these isolates. And from this, we were able to both reconstruct the population history for how these isolates related to one another. We were able to annotate the genes in these genomes and begin to understand which genes exhibited presence-absence polymorphisms and which genes were evolving between the isolates. And then also, because we have live cultures for these several thousand strains, we could then begin to do genetics on them. So our first question when addressing this system was whether we saw any Pseudomonas species or subspecies that were abundant across populations or reaching high frequencies within populations. So here I'm showing you a neighbor joining tree from the 16S analysis of the Pseudomonads that we found across all of our samples. And what was immediately evident, there's this specific OTU or subspecies, OTU5, that was enriched in the endophytic compartment. So that is inside of the leaf. It was present outside the leaf, but it was enriched inside of the leaf. It was found in approximately 50% of the plants, but at varying levels. So some plants had very low levels, others have very high levels of this operational taxon unit, and it was found at every time point. Now, Pseudomonas is not always a pathogenic genus, and actually some Pseudomonas strains exhibit biocontrol properties. And so just seeing the presence of a subspecies of Pseudomonas in our populations was not telling us that there is a pathogen present. So to assess how members of this clade affected the growth of the plant, we designed and implemented a very simple axenic assay, which means in the absence of other microbes, in which we took a single genotype of Arabidopsis and we colonized it with different isolates from the field, and we saw how well they grew. As a negative control, we treated just with buffer. As positive control, we treated with a known bacterial pathogen. This is a Pseudomonas syringae, DC3000, and our readout was the growth of the plant 1 week after treatment. So consistent with positive and negative control, we see that treatment with the pathogen produces much smaller plants after treatment. When we took isolates of Pseudomonas that were not classified as OTU5, just classified as various other subspecies, we found they showed a range of phenotypes, but only 10%, I'm showing you a fraction of the data here, but only 10% of them significantly reduced the growth of the plant in comparison to the buffer. When we took isolates classified as OTU5, we found the opposite. We found the converse. We found that 90% of the isolates that we tested classified as OTU5 significantly reduced the growth of the plant, and several of them outright killed the plant within 2 days of infection. Now, what these results suggested to us is that when we look at a plant microbiome and we see that OTU5 is abundant within the plant, it's suggestive of some level of infection, of pathogenic infection. But what the results also showed us is that these isolates, which were classified at the OTU level as being the same subspecies, were phenotypically different from one another, suggesting that they were genotypically different from one another. So this got us back to our original question. in trying to understand whether the pathogens we see in our Arabidopsis are basically a clonal expansion, or instead whether there's greater genetic diversity. So to give you just a little bit of perspective on this, a recent outbreak of Pseudomonas in kiwi performed dating of the time to the most recent common ancestor of these kiwi isolates. And their dating gave an estimate of somewhere between 30 to 80 years to the time to the most recent common ancestor. So this is a clonal expansion across these kiwi populations. So here we used the core genome phylogeny from the full genome sequences of our Pseudomonas OTU5 here, and we asked how divergent from one another they were. And we found over the core genome phylogeny, they averaged about a 2 to 5% sequence divergence at the nucleotide level. And if we recalibrate this according to the estimated substitution rates from other Pseudomonas, this comes to a time to the most recent common ancestor, something like 300,000 years. So this is a rough estimate, but it is orders of magnitude larger from what is known from many of the agricultural outbreaks. So what this is suggesting to us is that these pathogens have been localizing and no strain has overtaken in the recent history. And instead we have this long-term maintenance of this pathogenic diversity. Now, not only do we get different isolates colonizing co-localized plants, but actually if you look within a plant, you see that multiple genotypes will colonize even a single plant. So each phylogeny you see here is the phylogeny of all the isolates collected from a single plant. And you see, even within a single plant, you have genetic diversity of the Pseudomonas that are colonizing that plant. So what these results suggested to us is that in contrast to the paradigm that we see so frequently in agriculture, in which single lineages emerge and spread widely to be overtaken by another single lineage that emerges and spreads widely, at least in these Arabidopsis, in the Arabidopsis Pseudomonas interaction, we see no evidence of single lineages emerging and spreading widely. We see pathogen actually rather frequently, but instead we see this long-term maintenance of pathogenic diversity in these populations. So what is it that's preventing single lineages of pathogen from spreading across these populations? In the plant pathology community, there's been a large focus on this idea of pathogen-host genotypic trade-offs. And that is to be able to colonize one host genotype comes at the cost of colonizing another host genotype. And indeed, there's a lot of molecular evidence to support how these trade-offs actually can work at a mechanistic level. Another is that of environmental trade-offs, which I alluded to before, which is colonizing a micro-niche in one part of a field comes at the cost of being able to survive very well in another micro-niche. And such that because of the heterogeneity in the populations, you get this maintenance of diversity over time. So what we've embarked upon now is trying to understand what enables the pathogenicity of these OTU5 isolates in our Arabidopsis populations and which genes are evolving to generate the diversity that we see. And in the second part, which I'll talk about very briefly, is our first attempts to try to see how different environmental variables may be associated with pathogenic spread. So to try to understand which genes and genetic changes are underlying the differences in pathogenicity we observe, we're looking to the pan-genome of the Pseudomonas isolates that we sequenced. So many of you may know this, but bacteria have very labile genomes in which they lose and gain genes very quickly, meaning that even if you compare two E. coli, two strains of the same species, you can find that they differ, or they share rather, only about 70–80% of their genes. And instead, they're losing and gaining via plasmids and other mechanisms of horizontal gene transfer, large swaths of even hundreds of genes. The result is the picture that I show you here. So this is the pangenome. These are all of the genes observed across the isolates that we sequenced. This is the maximum likelihood phylogeny of the 1,500 isolates that we sequenced, and this is a presence-absence matrix for genes that we annotated in these genomes. If it's dark blue, each column is a different gene. If it's dark blue, it's present. If it's light, it's absent. So what you see here is these are all OTU5 isolates, and these two clades, which are closely related to one another, have these groups of genes that are conserved in one clade and absent in another clade. And this is within OTU5. But perhaps the most striking observation here is that there are about 600 genes that are largely conserved or at least enriched in OTU5 and largely absent outside of OTU5. And so if we want to understand what enables OTU5 to be pathogenic, it's reasonable to hypothesize that some of these genes may contribute to the phenotype. Now, we know a fair amount about the molecular biology of how Pseudomonas pathogenizes its hosts. And so we can specifically annotate genes that we know previous studies have identified as being involved in pathogenicity. And when we annotated a swath of effectors or pathogenicity factors, we found that there was a single effector called AVRE that seemed to be largely conserved across OTU5 and largely absent outside of OTU5, which makes it a good candidate for the transition to pathogenicity in this clade. We did a knockout test to see if indeed we could restore the growth of the plants if we knock out AVRE. Here on the y-axis, I use the metric of green pixels, which is just a digitized metric where we parse these images and count the number of green pixels versus in a dead plant. And when we knock out AVRE, we see that we get a significant restoration of the growth of the plant, which is suggestive that AVRE is at least in part responsible for the transition of OTU5 to pathogenicity. However, you may remember that we saw significant phenotypic variation even within that subspecies, in the subspecies in which AVRE is largely conserved, suggesting that there are other genes that are contributing to variation in pathogenicity. So to try to understand both how pathogenicity evolves along the phylogeny or within the phylogeny, and also to understand which genes are contributing to this, we are combining a screen across strains and a functional genomic screen within a single strain. So to try to understand how the trait varies within OTU5, we took 92 different isolates. We did infections as I described before. We did the image parsing that I mentioned to you before, where we took pictures of all of our plants, we parsed the images, we counted the number of green pixels. If there are more green pixels, it's a healthier plant. And through this high throughput method, it allowed us to look along the phylogeny to see how our trait changed between isolates. So what we saw, consistent with what we saw before, so here on the x-axis, this is the number of green pixels. These are healthier plants if they're over here, and they're basically dead if they're down here. And this is the mean of 8 to 10 replicates. So we saw before that plants that are not microbes that are not OTU5 have less of an effect on the plant growth. But within OTU5, we see what look like taxonomic shifts. So you see this clade here tends to be less pathogenic than it's an adjacent clade here, which actually most of the isolates kill the plant within a few days. So as I said before, we know AVRE is broadly conserved across OTU5, which is suggestive that perhaps it was gained here in the phylogeny. But we don't know what distinguishes this clade from its neighboring clade. And actually, we know that there are hundreds of genes that differ between these two clades, and which of them are the ones evolving and contributing to the differences in pathogenicity is not yet clear. So to try to narrow our candidate lists in a high throughput fashion, we've conducted a transposon mutant screen using the TN5, which inserts semi-randomly in Pseudomonas. We're able to generate, we've done this twice now. This is one mutant library with 25,000 mutants, another with 100,000 mutants. You take a single isolate, you mutagenize it with TN5, it inserts in every cell in a different place. And then you track the frequency of these mutants. So it inserts and putatively knocks out the function of the gene in which it inserts in. And then you track the frequency of these different mutants after infecting a plant. So here in this cartoon, you see that there are many different mutants. After infection with the plant, you see only the red and the blue seem to come out in the end, which is suggestive that either this was very advantageous to knock out the genes that were knocked out in these 2 isolates, or these other ones, it was deleterious to knock those out and they could not survive well in the plant. So as a first picture of the results we're getting, I'm showing you here a volcano plot, which is tracking the change in the frequency of these different mutants over time. So these ones over here are putative advantageous loss of function. The ones with an FDR less than 1% are in green here. So these ones are putative advantageous loss of function, where we see them increase in frequency upon colonization. These are deleterious loss of function. And from this first blush, from these first analyses we've done, we see that somewhere between 20 to 30% of the genes seem to be important for survival in planta. And what we're doing now is overlaying these data with what we know about the evolution of these genes along the phylogeny and which ones correspond to the pathogenicity shifts. But perhaps the newer data for us right now is our analysis of what's dictating the abundance of OTU5 over environmental conditions. So Arabidopsis, as I mentioned before, is almost globally distributed. It's found in very different locations, from Northern Europe, from wet, cold climates, to Southern Italy, where it's very dry. And in collaboration with several dozen other people in my previous laboratory, we went across Europe and collected Arabidopsis, its microbiomes, and Pseudomonas from over 300 populations of Arabidopsis from these very different environments. And from each of these collections, we garnered information about plant genotype. We did shotgun metagenomics, information on plant microbiome, we collected Pseudomonas, and then also acquired climate and weather variables. And I can't talk about this project without talking about these 2 women who, this was the lab manager from my previous lab, and this is, a technician with whom I worked very closely. And so I'll just show you one figure today from this project so far. But one of the first questions I had for looking at these data was looking at the distribution of the pathogen for which we knew a lot about in Southwestern Germany, and that was OTU5. I was curious to see if it was locally restricted. We see the evolution of these pathogens seems to be pretty rapid, and that there seems to be a lot of genetic diversity. And so I was curious to see if it was found throughout Europe, and also if we could see if it was associated with any environmental variables. So here I'm showing you the distribution of OTU5 in the microbiomes of these over 300 populations. And what we found was that OTU5 is widely abundant. We find it in these southern regions, but at very low frequencies in the microbiome, with higher abundance in the more northern regions. I want to point out that what I've shown you today in the population genomic study is actually just focused in this small little bubble here. And even within that small little bubble, we see what is to me extensive genetic and phenotypic diversity. And so looking at this picture, it's interesting for me to think about if we see this dynamic at play in all of these other little putative hotspots, or whether there are certain conditions that lend themselves to other dynamics, even for the same host pathogen system. So in conclusion, our first study, I think, demonstrated that Arabidopsis does not seem to be colonized, at least in Southwestern Germany, by a clonal expansion of Pseudomonas. Instead, we see pretty extensive genetic diversity and phenotypic diversity, even within this small region. We identified a single pathogenicity factor that seems to be responsible for a switch to pathogenicity, but there are likely dozens to hundreds of other genes that contribute to the evolution of this trait. And lastly, we're beginning to get a picture of the distribution of OTU5 over space. And I think intriguing for me to think about what's causing the hotspots for OTU5 and which environmental factors, biotic and abiotic, may dictate this distribution. So the project I talked about today was conducted, or the projects, at the Max Planck Institute in Tübingen, Germany, in the laboratory of Detlef Weigel. I worked very closely with Manuela Neumann, and a lot of this would not have gotten done without her. I have close collaborators, Richard Nayer, Joy Bergelsen, and Eric Kemen, and various other institutions. But before I let you go, looking forward, I continue to be interested and want to pursue these questions of what is limiting the spread of pathogen within and between host species. And I plan to pursue these questions using a combination of field collections, population genomics, and genetic analysis. So if this interests you, I would be interested in talking to prospective graduate students and undergraduate students. We have computational projects, field projects in conjunction with computational projects, as well as molecular population genomics projects also. So if you're interested, you can check me out. This is my website, and shoot me an email. And thank you for your attention.
Thank you, Talia. That was a beautiful presentation. Crystal, did you want to read these questions to Talia that have shown up now in the chat?
Yep. So I have a question from Jack asking, does Pseudomonas itself have pathogens?
You mean phage or something of the sort? Yes. So phage is very extensively studied in Pseudomonas aeruginosa, which is a somewhat close relative of the Pseudomonas I study here. And there's been a little bit of work on phage in syringae relatives, which is the major focus here. So the answer is yes, though it is understudied.
Okay. And a question from Richard, doesn't the data just suggest an Asian clonal expansion relative to agriculture? And any ideas about the initial expansion for O25, for example, like population restrictions in an ice age?
Yeah, an ancient clonal expansion, right. So the transition of OTU5 to pathogenicity, and then it expanded widely. Yes, I agree. So that would be a good way of framing it, is that it's an ancient clonal expansion for which there was no overtaking of the next generation. It was persistent divergence, persistent survival of the diverse lineages.
Okay. And that's all the questions people have posted so far.
I have a question for you, Talia.
Mm-hmm.
So you say there are about 600 genes that were common to that OTU5. What other things do you speculate to find? Do you think many of those are related to pathogenicity?
Yeah, so the genes I think that are coming up the most commonly now are things like siderophores, so genes involved in iron acquisition. And so I think that the classic pathogenicity factors, which are effectors, which are interfering directly with the plant immune system, those ones, they don't seem to have many of those kind of classic effectors. But when it comes to dealing with nutrient acquisition and sequestering, I think that those are probably going to be the most common looking forward.
So do you think then that stealing iron from the plant, is that highly detrimental? Do you think that's contributing to their pathogenicity in some way?
I think, you mean whether the resource is limiting for the plant itself, the amount that it's stealing. I think it's probably more the ability of the plant to limit the proliferation of the microbe itself. And so we find there's a very tight correlation just between how many microbes are there and how little growth there is for the plant. And so probably if the limiting resource is iron itself for the microbe, it's not that the plant is iron limited, it's that the microbe can then proliferate to the point where then it's also acquiring the carbohydrate based resources.
Actually, there are two more questions coming up. One from Dean. For genotype trade-off, you expect GXP interactions. Most of O205 pathogenicities are in a single Arabidopsis genotype. The question mark. And so other strains might be pathogens with other genotypes.
Yeah, so we do see genotype. So I'm not showing this today. That's a graduate student in the lab. She started doing these G by G matrices, and you see that you see changes in the rankings of OTU5. So that it's pathogenicity AVRE seems to be broadly pathogenicity inducing, so that one factor, but then the others seem to be genotype specific.
Okay. And one question from William. The strains that don't show pathogenicity, what are they doing instead? Infecting other organisms?
Yeah, unknown. So there's been some work that shows some amount of cheating so that they can co-colonize with the pathogenic strains, and that when they co-colonize with the pathogenic strains, they actually can out-compete them to some extent. And then others just may be environmental microbes that just get in through the stomata when the stomata are manipulated to be open.
Okay.
Well, thank you. Thank you, Crystal, for your help with these questions. And thank you, Talia. This is really very interesting talk. Can you join me in thanking Talia?